Okay, welcome to part 2 of the chapter 7 lecture. We're continuing with photosynthesis, and as I was saying, there was an experiment back in the 1800s where Engelmann observed that oxygen-dependent bacteria would congregate in a certain area near algal cells. Okay, so that was the experiment that he set up. The results were that bacteria mostly congregated around algae exposed to red, orange, and blue-violet light, and they rarely moved to areas of green light. His conclusion was that chloroplasts absorb light mainly in the blue-violet and red-orange part of the spectrum. So remember that oxygen production is an indication of where photosynthesis is occurring, since oxygen is a byproduct. So because these bacteria were congregating in areas where the algae was exposed to red, orange, and blue-violet light, he concluded that the chloroplasts were mainly using those wavelengths of light for photosynthesis. And here's a depiction of what that experiment looked like. He had a microscope slide here with a string of algal cells as well as bacterial cells on there. And if you were to look at that microscope slide under a microscope, this is what it would look like. So the number of bacteria is here on the y-axis, with more bacterial cells being up here and fewer being down here. And then the wavelength of light in nanometers is on the x-axis here. So he observed that there were not really any bacterial cells in this area of green light. This is called a bimodal distribution because there are two areas of the graph where you have a lot of observations of your bacteria, so it kind of forms two mountains here. Okay, so a little bit more about chloroplast pigments. Chloroplasts contain several different pigments. There's not just one type of pigment, there are several. Two of the most common are the chlorophylls A and B. Chlorophyll A mainly absorbs blue, violet, and red light and participates directly in the light reactions. And chlorophyll B absorbs mainly blue and orange light and participates indirectly in the light reactions. Carotenoids are another type of pigment. They absorb mainly blue-green light. They participate indirectly in the light reactions. And they absorb and dissipate excessive light energy that would damage the chlorophyll pigments. And carotenoids are ultimately responsible for the spectacular colors we see in the fall on trees. So they are due to the yellow-orange light that's reflected off of the carotenoids, and the falling autumn temperatures is what causes a decrease in the levels of the primary pigments, which are chlorophyll. And that allows the colors of the longer-lasting carotenoids to be seen in the leaves where before we wouldn't normally see them. Light behaves as waves, but it can also behave as photons, which are fixed quantities of light energy. And that is really the raw material with which chlorophyll molecules work. They absorb photons, and electrons within those pigments gain energy as a result. As the electrons fall back to their ground state, energy is released as heat or light. This is one of the reasons why really dark surfaces like black or very dark gray pavement gets very hot in the summer, because the electrons in those pigments are falling back down to their ground state after light strikes them, and they're releasing heat as a result. And this is also ultimately why glow sticks glow. So some photons will cause electrons to release just heat, but some types will also release light. So fluorescent dyes have the type of electrons that will release light in the form of fluorescence due to this reaction. So here we have a chlorophyll molecule, which would be found within a chloroplast. Light energy in the form of a photon hits that chlorophyll molecule, and as a result of that, we have an electron within the pigment that goes into an excited state. This excited state is very unstable, so typically what happens is that electron falls back down to its ground state almost immediately. It releases heat, in some cases it releases light as well, and then the ground state here can be thought of as the starting point. So how exactly do photosystems harvest light energy? Well, in the thylakoid membrane, there are chlorophyll molecules that are organized with other molecules into photosystems. And a photosystem is a cluster of a few hundred pigment molecules that function as a light-gathering antenna. So they are basically just functioning to gather those photons. When a photon strikes a pigment molecule, energy jumps from one molecule to another until it arrives at the reaction center within the photosystem. The reaction center of the photosystem consists of chlorophyll A molecules that sit next to another molecule called a primary electron acceptor, which traps the light-excited electron from chlorophyll A. Another team of molecules built into the thylakoid membrane then uses that trapped energy to make ATP as well as NADPH. Here we have a depiction of a chloroplast again with stacks of thylakoids, and these photosystems are present within the thylakoid membrane here. So this is a close-up depiction of a thylakoid membrane, similar to a plasma membrane of a cell with that lipid bilayer. We have the hydrophobic tails and the hydrophilic heads. And there's a cluster of pigment molecules that spans that membrane, and this is where that photosystem is located. Whoops. Okay, so if we were to look at the top of this membrane spanning photosystem here, we have a cluster of pigment molecules on top, and what happens is a photon strikes one of these. There's going to be a transfer of energy from one to the next until it reaches the reaction center, which consists of the primary electron acceptor here, and electron transfer occurs between these pigment molecules and that primary electron acceptor. How do the light reactions generate ATP and NADPH? Well, there are two types of photosystems that cooperate in the light reactions, and there's the water-splitting photosystem as well as the NADPH producing photosystem. What happens is photons excite electrons in the chlorophyll, and those electrons are then trapped by the primary electron acceptor. The photosystem replaces these electrons by extracting electrons from water. This is the step that releases oxygen. So this reaction center needs to replace those electrons that are transferred to the primary electron acceptor. So to do that, it strips electrons from water, and because of that, hydrogen ions and oxygen gas are released. After this initial primary electron acceptor grabs an electron, those energized electrons pass down an electron transport chain to the next photosystem. And because of this electron transfer, there's going to be energy available to make ATP. Chloroplasts use the energy from the fall of electrons to make ATP. There wasn't quite enough room to fit all of this depiction or illustration on one page, but this is that same electron transport chain here, and this is a second reaction center in a second photosystem. So this is the NADPH producing photosystem, which transfers its light-excited electrons to NADP+, which reduces it to NADPH. This is another way of looking at that with these two photosystems here, and that is going to transfer the raw products there, which are NADPH, as well as ADP plus into the Calvin cycle, which I will cover next. Okay, so the Calvin cycle is what actually makes sugar from carbon dioxide. It functions like a sugar factory within a chloroplast, and it regenerates the starting material with each turn. Okay, so first of all, we have to have CO2 present from the air for this to work, and an enzyme adds carbon dioxide to a 5-carbon sugar called RUBP. The resulting molecule breaks into 2 3-carbon molecules, and one of those resulting molecules is shown here. With energy from the ATP and NADPH, which were provided by the light reactions, enzymes convert each 3-carbon molecule to the 3-carbon sugar G3P. For every 3 molecules of carbon dioxide that enter the cycle, the net output is 1 G3P sugar. The 3 in G3P just refers to the fact that there are 3 carbons in it. And from this G3P sugar, glucose and other organic compounds needed by the plant can be produced. Finally, using energy from ATP, enzymes rearrange the remaining G3P sugars in order to regenerate RUBP, so the cycle can start again. And interestingly, there are some adaptations for certain types of plants that live in really dry environments where they have to close their stomata and conserve water that way. So the common plants that are very widely distributed are known as C3 plants. And the 3 again refers to this 3-carbon molecule that's used in the Calvin cycle. These plants use CO2 directly from the air, so they have to keep their stomata, which are those tiny pores, open in order to gather CO2 from the air and complete photosynthesis. But what if you have a really dry environment where plants need to close their stomata in order to prevent water loss through evaporation? And basically have this trade-off between trying to conserve water and taking in enough carbon for their photosynthetic reactions. Well, we have 2 different evolutionary solutions to that problem. One is a C4 plant, and C4 plants close their stomata to save water during hot and dry weather, and they can still carry out photosynthesis. And CAM plants are adapted to very dry climates, such as deserts, and they only open their stomata at night to conserve water. Your book has a lot more information on these plants and the modifications that they have in order to complete photosynthesis, but I don't need you to know that. That's just extra information if you'd like to learn more about those plants. But as an example, we have the C4 pathway. An example of that is sugarcane, another is corn, and then the CAM pathway plants such as pineapple, and then succulents and cacti are also CAM plants. Okay, that timing worked out really well. This is the end of the Chapter 7 lecture. Again, students, if you have any questions, please feel free to email me. and use that peer-to-peer discussion board.